Welcome to my last and final video from me. Now, always keep in mind that if you have anything or you are confused about anything, you comment down below and me and my friend Stephanie will hopefully answer to your question as soon as we can. So now we're going to talk about cell cycle. This, my friend, is a cell cycle. Oops. It's upside down. This is a cell cycle. Right here, that's where the juice happens. This is interphase. As you can see, interphase is much bigger than prophase, metaphase, anaphase, and telophase, and cytokinesis. Which is why interphase, like most cells are in their interphases most of the time. So, let's talk about it. So in G1, that's when the cell grows and matures and grows whatever it needs to grow. And S, it's when DNA replication happens. We'll talk about DNA replication in a bit, by the way. G2 is the preparation for division. So it's getting prepped before it gets to prophase, metaphase, anaphase, and telophase, and cytokinesis. Basically, the cell division is getting ready for it. Is getting ready before it gets to the cell division. G0 is the resting phase. This is where they can chill and be like, yo, Wyatt, like, what's up, bro? Or, I don't know, whatever they do. But this is their resting phase, but obviously they come back. This obviously whole thing results in creating two daughter cells. By the way, this is like a little graphical, situation. Well, to me, this is like the visual, but we'll talk about that one in a bit. We'll talk about mitosis in a bit. So, once it gets to the prep for division, it gets to prophase. What is prophase, you may ask? Prophase is when it lines up the chromosomes. It's like when it's getting ready to, like, it's lining up the chromosomes. It's not lined up yet, it's getting the chromosomes to line up. Which means the spindle fibers are being activated in this situation. Now, when it gets to mitosis, why did I just say mitosis? I meant to say metaphase. I can't think straight. Metaphase, that's when it literally lines up. That's when the chromosomes are in a straight line and the spindle fibers are connected to them in every, little way. Now, anaphase is when the real action happens. That is when the chromosomes are being separated by their base. They're being no. It's like Jack and Rose, I guess you can say, from Titanic. They're being separated by the spindle fibers. So the spindle fibers are pulling them apart and apart on the opposite ends of each other. Telophase is like when they're halfway splitting apart, which creates a cleavage furrow. Because it really looks like a cleavage. And that's when the nucleus reappears, chromatin reappears, and now there's only one centrals on each of the cells. But the centrals are obviously on the opposite end of each other. Everything is. Now, cytokinesis is when it results in two daughter cells, and that's when they fully separate, and ta-da! The daughter cells are created. So that's basically the whole cell division. So this is a mitosis stage. I did not create this paper, obviously. This is too organized to even be my paper if I even did create it. So I labeled it for you because they were a little bit scrambled since I had to find the order myself. And with a group of help, obviously. Now, an interphase You can see there's a nucleus, there's a chromatin, and obviously there's that one centriole. Once it gets to the real action, that's prophase. Now there are two types of prophase. There's the early and there's the late. Obviously, early goes first and late goes second. But basically what the early is the nucleus disappears and the spindle fibers are being activated. Now on prophase, late, that's when the spindle fibers are like shifting and putting the chromosomes in a line. Putting them, it's not fully in line yet, they're just putting them so they can get ready for metaphase. Oh no, you clicked on that. So metaphase is when they are really lined up. As you can see, the spindle fibers are connected to them. And that's all I can say. Now, when you get to anaphase, this is the Jack and the Rose. They get separated by the spindle fibers. So the spindle fibers actually separates them, like it pulls the chromosomes apart in half, and one half goes to the left, and the other half goes to the right. Now, telophase. What is telophase, you may ask? As you can see, this is what I mean by it creates a cleavage furrow. It really looks like a cleavage. But the nucleus appears, it reappears on both. There's also a chromatin that appears in both. But now, the centrioles are on the opposite ends of each other. Now, cytokinesis, as I said, it just results in creating two daughter cells. Now, let's go. Since we're done with mitosis, let's go to meiosis, shall we? So I am going to use my notebook just because it's a little bit detailed, and I feel like my teacher, Ms. Downey, helped out. Shout out to Ms. Downey. But now, metaphase is different from mitosis because meiosis has an extra space, so it has prophase 1, prophase 2, metaphase 1, metaphase 2, anaphase 1, anaphase 2, and all that. So let's start with the first interphase. Obviously, it starts with 2N. It starts with 2N. It all starts with 2N until it gets to telophase and cytokinesis. That's when it goes to 1N. But I'll tell you later. So in the interphase, that's when the growth, development, the DNA, replication, and the prep for cell division happens. The prophase 1. The nucleus dissolves, spindle fibers form, chromosomes form, homologous chromosomes forming, what does that say? Form tetrads. And also, keep in mind, prophase is when crossing over occurs. And do you know what crossing over means? It means like genetic variation. Okay? That's what crossing over. Now, metaphase 1. Homologous chromosomes line up in pairs. So you can see right here, I don't know if I should zoom in just because. There you go. Whew. As you see, crossing over did occur. And now they have different color. This has orange and purple, orange and blue, blue and orange, purple and orange. That's because right here, that's when crossing over happens. I think I'm going to stay in this view actually. You can probably see a little bit better. So now let's go to anaphase 1. Homologous pairs separate. Pairs. So in mitosis, they literally separated from their base. But in meiosis, they separate from pairs. So as you see, there used to be 4 pairs. Now there's 2 pairs on each one. Telophase 1. As you can see, it stays on 2N. But once it gets to telophase 1, there's only 1 N. Nucleus reforms. Chromosomes decondense. And division pauses for quality control. So now it creates 2 cells with obviously the different colors because crossing over. But the action doesn't stop there. Oh, no, no, no. Prophase 2. It's now this whole thing will stay as 1N just because it's not going to be in pairs anymore. Nucleus dissolves, chromosome forms, and spindle fibers are activated. Well, bro. So it's like, basically it's almost like this, what just happened on top, but just it's repeating again on the bottom with just 2. Metaphase, chromosomes line up in the middle. The chromosomes pairs line up in the middle, as you can see. The spindle fibers are attaching to them both on either sides. On anaphase 2, the sister chromosomes separate. Now this is when it looks like this right here. That's when it looks like this. They don't separate in sisters like on what just happened on anaphase 1. They separate like, you know, like before. I messed up a little bit right here, but that's okay. I'll go here and here. On telophase 2, the nucleus forms and the chromosomes decondense. So now, usually it would just be 2 cells. But guess what? It's 4 haploid gametes. Gametes? See? And they are different. These are not the same at all. Each one is different because crossing over happened, and crossing over means genetic variation. So now, let's talk about DNA replication. Bam! So I did zoom in a little bit just so you can see. Hope you don't mind. So, this is the DNA replication fork because it literally looks like a fork. Like, la la. So this is a DNA strand or a parent strand. This is the parent strand. It's still the parent strand no matter what, even if it's separated, because at the end they come back together just like this. This is the helicase. This is the zipper that unzips the DNA as it goes on. Now, the way this starts, I don't think you can see. Right here, how I remember it, the leading strand will always be the long strand. And there's always going to be a box at the end. I always remember the box is 5 prime. Meaning, at the bottom, oops, sorry, you can't see it. Meaning, at the bottom, it'll be 3 prime. And since this strand, parent strand, is 3, the opposite, or parent strand, is the 5 prime. And then the below is the 3 prime. So it literally goes opposite from each other. Now, the leading strand, you will always know when the leading strand is if it has a long line, and the box will always be at the end. Also, keep in mind, the only way to know, the box will always start with 0. I mean with 5. I'm so sorry. With 5. What I mean is, as you can see on the lagging strand, there's a 5 here on the box, 5 here on the box, and 5 here on the box. Obviously, at the opposite ends, there's a 3. Now, the little strands right here are called Okizagi fragments. Hope I said it right. Probably didn't. But yes. The 5 will always be on the box. And they actually help because if you're when you're finding the order of this, the 5 and the 3 end, it will be helpful. And yeah. This is all for today. I really hope you enjoy it so much. Thank you for watching. Thank you for keeping up with me actually and dealing with my bad, pronouncing things. But yeah. Hope I see you again soon. And I will see you whenever I can, I guess. Bye.